Welcome back to a brand new explainer. Let's jump right in, because today we're looking at something literally foundational to human life. I want you to picture the human body, not just as, muscles and bones, but as the most sophisticated, microscopic cellular assembly line in the universe. Right now, in millions of your cells, there's an engine running constantly. It's synthesizing DNA, repairing tissues, building life itself. And today, we're going to decode the blueprints of that exact engine by looking at the science of folate, choline, and the unbelievable mechanics of human development.
Which brings us to a pretty high-stakes question. What does it actually take to build a human brain? Think about it. It's not just a matter of giving it enough time. It requires a highly specific set of raw materials, and they have to be delivered at exactly the right moment. The central nervous system of a fetus is essentially a rapidly expanding universe of dividing neural cells, and it absolutely relies on critical fuels, specifically folate and choline. If these biological supply chains get interrupted, well, neurogenesis, neuronal migration, synaptogenesis, the whole shebang can just derail. So how exactly does this biological supply chain operate?
Okay, let's dive into this with Section 1, the folate pathway explained.
To understand how we literally build a human, we have to look closely at the one-carbon metabolism network. It's the central biochemical hub delivering methyl groups for DNA synthesis, amino acid transformations, epigenetic regulation, really important stuff. And at the absolute heart of this pathway is a brilliant piece of microscopic machinery. It's an enzyme called dihydrofolate reductase, or DHFR. It's a conserved enzyme made up of exactly 187 amino acids, structured in what scientists call an alpha/beta fold. You can kind of think of it as a specialized factory press. Its main job? To take a molecule called dihydrofolate and convert it into active tetrahydrofolate, which is absolutely crucial for creating purines and thymidines. Without those, there is simply no way to make DNA or divide cells. But how does this tiny machine actually do that?
The mechanics here are honestly mind-blowing. The DHFR enzyme has an active site featuring this highly flexible lid. And this lid is made up of three specific protein domains, the MED20, the FG, and the GH loops. When the enzyme gets to work, this lid mechanically closes right over its cofactor, NADPH, and the folate substrate. It literally physically encloses them, stabilizing their interaction to catalyze a chemical reaction through a stereospecific hydride transfer. I mean, it's a precise, physical clamping motion that is happening millions of times a second inside you right now.
But that leads us to Section 2, when the engine breaks.
So what happens when that specialized factory press just breaks down? What if a genetic mutation damages that flexible lid or stops the enzyme from working altogether? Well, I want you to look at this number, 20. Based on recent scientific literature, there are fewer than 20 reported cases in the entire world of a genetic condition called DHFR deficiency. It's an ultra-rare autosomal recessive disorder. Because the DHFR enzyme is so fundamentally critical for basic cellular survival, individuals who inherit biallelic pathogenic mutations in this gene face incredibly severe, life-altering consequences.
The absolutely crucial point here is understanding the cascading effect of a broken engine. The symptoms we're looking at are devastating. See, because DHFR is necessary to recycle tetrahydrobiopterin, which is a cofactor you need to synthesize neurotransmitters like serotonin and dopamine, patients experience severe neurological symptoms. We're talking developmental delays, microcephaly, and really severe seizures. On the blood side, we see megaloblastic anemia, where red blood cells get abnormally large and immature, or even pancytopenia, which is just a massive drop across all blood cell types. Plus, cerebrospinal fluid 5-methyl tetrahydrofolate is significantly reduced or even completely gone. The cellular assembly line just grinds to a halt.
Section 3, folic acid versus active folate. Now, if the cellular machinery that processes the fuel is broken, is there a way to bypass it entirely? Actually, yes. You just change the fuel. We hear a lot about folic acid in prenatal vitamins, but there's a vital distinction to make between folic acid and 5-MTHF, which is active folate. Folic acid is synthetic. To be used by the human body, it demands to be processed by that exact DHFR enzyme we were just talking about. 5-MTHF, on the flip side, is the predominant natural, biologically active form of folate. It doesn't require any enzymatic reduction by DHFR whatsoever. It's basically a ready-to-use biological fuel. And this brilliantly illustrates the real advantage here. Because 5-MTHF doesn't need to fight its way through the metabolic bottleneck of DHFR conversion, it boasts far better bioavailability. It gets directly absorbed and is immediately available to the cells.
And that is incredibly important when we start looking at real-world human genetics. Because as it turns out, mutations in the folate pathway are actually pretty common in the general population. Specifically, we see mutations in the MTHFR gene like the C677T and A1298C variants. If a woman has these mutations, her body is really going to struggle to convert synthetic folic acid into the active form. This can lead to folate deficiencies even if she is diligently taking folic acid supplements, which obviously increases the risk for neural tube defects. So for these individuals, supplementing directly with 5-MTHF bypasses the enzymatic blockage entirely. It ensures the developing fetus gets exactly what it needs without relying on, inefficient cellular machinery.
Moving on to Section 4, choline and the brain.
Let's see how all this builds into another completely essential, but honestly often overlooked component of the prenatal engine, choline. While folate gets a lot of the spotlight, and rightly so, choline is an absolute powerhouse for fetal brain neurogenesis. Choline is required to synthesize phosphatidylcholine, the main structural component of cellular membranes. It's also the direct precursor to acetylcholine, the neurotransmitter heavily involved in learning, memory, and concentration. The European Food Safety Authority suggests pregnant women need about 480 milligrams of it daily. We get it from sources like beef liver, eggs, and lean meats. But here's the alarming part. Most women of reproductive age actually fall way short of this intake, putting optimal cognitive development at risk.
And it's not just dietary gaps, though obviously vegetarian or vegan diets that miss out on animal-based choline sources are a factor. There are specific risk factors that really exacerbate these deficiencies. Lifestyle factors like smoking and alcohol consumption actively deplete choline concentrations in the body. Furthermore, just like with folate, there are genetic polymorphisms, specifically in the CHDH gene, that can drastically increase an individual's biological need for choline. Let's pull this all together in Section 5, Tailoring Prenatal Supplementation.
So knowing everything we now know about the complex, interconnected machinery of folates, DHFR, MTHFR mutations, and choline, how do clinicians actually apply this? Well, human biology is dynamic, so our approach to fueling it has to be dynamic too. And that brings us to a highly tailored clinical intervention timeline. A pregnancy isn't just one static event, right? The nutritional demands shift dramatically as that cellular assembly line scales up. During preconception, the focus is on universal folate, vitamin D, and iodine. Then, moving into the first trimester, active folate like 5-MTHF is prioritized, alongside vitamin B12, which works synergistically with folate and the 1-carbon metabolism network. By the second and third trimesters, the fetus is growing rapidly, so the demand absolutely spikes for structural and developmental elements like iron, calcium, magnesium, and DHA. And finally, postpartum supplementation needs to be specifically tailored to support lactation. Now, what's really interesting about this slide is when you look at the clinical entry point and decision columns here. This outlines a major shift away from a generic, one-size-fits-all multivitamin approach towards true precision medical care. Clinicians are now being advised to look closely at dietary history, test for specific deficiencies, like checking hemoglobin and ferritin levels, and make highly targeted decisions based on the exact reproductive stage and the unique genetic realities of the mother. And this need for a tailored approach is perfectly illustrated by the current clinical debate surrounding iron supplementation. On one hand, you have the World Health Organization advocating for routine prophylactic iron supplementation to universally combat anemia, which is undeniably a major global health issue. On the other hand, the American College of Obstetricians and Gynecologists recommends a screen and treat strategy. They argue that iron should really only be supplemented if a deficiency is confirmed, simply because unnecessary iron can cause gastrointestinal discomfort and oxidative stress. Both perspectives rely on the exact same medical science, but they apply it differently based on population needs versus individual precision. Which brings us to our final thought for today's explainer. We've looked at the DHFR enzyme, a microscopic lid opening and closing millions of times a second. We've seen how a single tiny genetic mutation can halt human development in its tracks. We've explored how active 5-MTH and choline literally build the memories and memory centers of the human brain. The science is incredibly clear here. Precision nutrition is fundamentally tied to human potential. So, considering the staggering complexity of this cellular machinery, are we doing enough to fuel the cognitive and physical future of the next generation? Think about it. Thank you so much for exploring this fascinating topic with me, and keep feeding your curiosity.